First of all, just thanks so much to the organizers for giving me the opportunity to speak here today. My talk will be changing directions a little bit and actually focusing on complex traits. And the motivation of our project is that complex traits are influenced by multiple genes. And in fact, in the underlying cellular mechanisms, these genes are often working together in intricate interacting pathways. Now, we'd often expect that this would result in genetic variants having non-additive impact on our phenotypes. For instance, the classic two-hit model in cancer progression, where there are two specific mutations required in combination to produce a tumor phenotype, and either alone would not produce it. Now, we know that epistasis and interactions are pervasive in model organisms, but we're not really sure yet what their impact is in complex human traits. But if we could find them, they would have the potential to offer significant mechanistic clues to complex disease and also to provide improved prediction of disease risk. So the goal of our project is going to be to detect these interactions from high-dimensional genomic assays. Now, the major challenge is that we're often statistically underpowered. So even if we're only testing pairs of genetic variants, we'll often have millions or even billions of tests. And we're also confounded by small effect sizes. So our real interactions are basically buried among a bunch of spurious false positives. And this sort of explains why we don't often see significant interactions reported in genome-wide studies. But I'm going to present a novel approach, which is based on a guided adaptive procedure, which is going to leverage gene networks and other significant patterns as prior knowledge to improve our ability to detect these interactions. So I'll describe just quickly our data and background before I move on to describing the method. Because we are curious broadly about the effects of interaction, I'm actually going to talk about three very different data sets using different assays and different traits. The first is glioblastoma survival time, which we obtained from TCGA. And in this case, we're actually going to use expression data and survival data for 450 subjects, 451. The second is a genome-wide association study where we have SNP data for about 3,000 subjects for type 1 diabetes. And the last is a drug sensitivity assay in cancer cell lines from the CCLE Cell Line Encyclopedia. And in this case, we're going to focus on using copy number variation as the assay. So we didn't end up doing anything particularly fancy for basic epistasis testing of each pair. So when we're looking for interactions, all we do is use a likelihood ratio test to evaluate whether the data suggests a deviation from an additive model. So in this case, we're really just looking to see whether adding a non-additive feature improves our ability to predict the phenotype using just a single pair of genetic factors. But one thing I will mention is that we do need to account for confounding factors. We'll get a lot of false positives if we don't account for things like age, gender, or broad trends from principal components. And in addition, one thing I'll point out is that we actually include nonlinear effects of individual genetic factors, because without those, we will in fact get several false positives. Okay, so the main thing that our project rests upon is that it turns out that interactions are not distributed at random throughout the genome, but rather they follow predictable and in fact biologically meaningful patterns. The first, and something that many people have used as a heuristic, is that variants with a marginal effect on the phenotype are more likely to yield interactions. So what we've done here in the drug sensitivity data is to bucket each pair according to the average significance of the individual gene. And then we look at enrichment for that bucket for lower p-values according to the interaction test. So what we see is that the stronger the effect of the individual variants, the more likely it is that the pair will show some evidence of interaction. Another major pattern that we investigated was network connectivity. So we have available to us now a significant amount of prior knowledge about which genes work together. We have protein-protein interaction information, co-expression, and other data. And what we can see, so one example I've shown here in glioblastoma is that using the string database for protein-protein interaction information, direct neighbors in this known network are in fact more likely, they statistically significantly are enriched for evidence of interaction. But that's not the complete picture. It turns out that indirect connections in these graphs matters too, and things like shared neighbors matter as well. And so what we did was to use a graph kernel, which uses a random walk to estimate the connectivity between every pair of nodes, even if they're not direct neighbors. So it takes into account things like strong connections due to multiple paths between two nodes. And if we look at enrichment for interactions among these pairs, what we see is that the stronger the connection, according to this graph kernel, the stronger the enrichment for interactions. In fact, when we look at our graph kernel, we get even stronger enrichment than just direct neighbors. One thing that I do want to note is that the effect of networks tends to vary a lot by the specific network we look at, and also by the trait that we're investigating. So this is another protein-protein interaction data set, and here we barely see any effect from direct neighbors, and this could be due to sparsity or just low coverage of the proteins related to this particular trait. And in fact, we get almost no significance at all from gene ontology in this case, but it does vary by the trait that we investigate. The final pattern that I'm going to mention is that in every trait we've looked at, there tend to be hubs of interaction. Now, these will be genes or pathways that have many interactions compared to random nodes in the network. And in fact, if you compare an interaction network with a randomly generated network, what we see is that we'll have many more genes with very few interactions and many more genes with many interactions. So it will look more like this compared to a random network, which just sort of has evenly distributed degree across the nodes in the network. And this is going to turn out to be a very important pattern for our method. So now I'll go ahead and turn to the method that we settled upon. I've already mentioned that if we were just to test all pairs blindly, we're going to be statistically underpowered in almost all of these high-dimensional genomic data sets. Another option, and something that people have looked at, is to pre-select a subset of genetic variants to look at. So they might consider only gene pairs which have a known protein-protein interaction, or only the very top marginal variants. But this can be restrictive, and as I mentioned in the network section, the choice that you make ahead of time might not be good for the trait at hand, and it's very difficult to predict this a priori. So instead, I'm going to propose a method that actually combines the patterns that I've discussed to robustly discover interactions. So our method is called Guided Adaptive Interaction Testing, or GATE. And the way it starts is that we come up with some simple heuristic prioritization. These are all the pairs of variants that we haven't yet tested. And what we're going to do is test only a very small subset of the highest ranked pairs. And we're going to use the results of that limited test and look at the attributes of those pairs. So we're going to look to see how they were connected among a set of known networks. We're going to look to see the identity of each of the genes and each of the pathways related to those variants. And we're going to look at their marginal effects. We're going to take all of these things into account into a predictive model in an active learning method where we actually attempt to predict the significance of each pair. And what we're going to do then is to learn this predictive model and use that to actually re-rank all of the pairs that we haven't yet tested, which at the beginning is most of them. And we're going to iteratively test the highest next ranked pairs, re-update our model, use those parameters to re-rank all the pairs that we haven't tested, and continue. So as we go, essentially, we hope that we're testing pairs in order of their prior likelihood of yielding some interaction. Again, so I'll be discussing results on the 3 data sets I mentioned, the glioblastoma survival time, type 1 diabetes, and drug sensitivity. So here's a standard result that we get from GATE. So on the x-axis is the number of pairs that we've tested. So as we go in our iterative procedure, we start at the very beginning and test more and more. And I'm comparing GATE to a model that just ranks pairs at random and one that ranks it purely according to the marginal effects of the individual variants. And what we see is that GATE offers dramatic improvement in the number of significant interactions identified. Now, there's 2 ways that one can use GATE. One is to actually stop early. So if we can heuristically detect when we've sort of reached this peak and we've tested all the things that are a priori likely to be interacting, we can simply stop. And this would directly reduce our multiple hypothesis burden and allow us to obtain more significant pairs. But the method that we usually use is actually to use a weighted FDRC commutation, which allows us to test the full set of pairs while maintaining significance. We get similar results in the drug sensitivity assay and in type 1 diabetes, in both cases significantly outperforming a random approach or one based purely on marginal effects. And overall, it's sort of striking that we do get a very large number of interactions for all 3 of these complex traits in very different assays. So I don't expect you to read all of these gene names, but what I wanted to point out is that hubs are a major driver of the GATE results. So you can see here that in each trait, we get a number of genes that have a very large number of interactions each. The median number of partners in each of the networks for these traits is about 1 to 2, compared to tens that we get for the hubs. And each of these is going to be highly weighted according to the predictive model that GATE learned. So I'll go into slightly more detail on a couple of the results. We noticed that for glioblastoma survival time, several of the top hubs were involved in TGF-beta signaling, which has been shown to be relevant to cancer progression, and a few of the genes are known to impact glioblastoma in particular. We also noticed that a few of them, and a few more down the list that I haven't shown, are related to lipid metabolism and cholesterol homeostasis, which has also been shown to be relevant in glioblastoma. But what's interesting, of course, is that we were able to detect non-additive effects between these two pathways, which could suggest some dependence of proliferation on lipids in glioblastoma. And the last result that I'll present is something that we found in type 1 diabetes. Now, in type 1 diabetes, the majority of the interactions that we pick up are purely within the MHC region of chromosome 6, where we already know that there are several individual risk variants, and in fact, epistasis has previously been confirmed in this region. But we did find a few outside of the MHC, and one that I'll highlight is between a SNP in the MHC region and a particular SNP on chromosome 1 that, when I first looked at it, was not near any genes that I thought looked interesting. But what I ended up finding is that it's directly proximal to a region of homology to mouse chromosome 4, which actually has a known type 1 diabetes risk locus for mouse. And this region had not been previously characterized for human. So that was kind of neat. So in conclusion, the message that I want you to take home is that if you're looking for interaction in complex traits, these interactions are not distributed at random, and you can use significant patterns, including known gene networks, marginal significance, and especially hubs in your traits, to guide the detection of epistasis. And our method that we present, in particular, adapts these patterns to the specific trait at hand, identifying relevant hubs and networks for the trait that you're interested in. And using this, we're able to discover a large set of interaction for diverse assays. And one particular result is the susceptibility locus that we found in human that was previously implicated in mouse. And I want to thank my co-authors, Hoon Cho, and my advisor, Daphne Kohler. Thanks so much for your attention.
Questions?
Thank you very, very much for a very nice talk. 2 questions. So the first one is about that little blip right before you fall down. So the reason for the blip is that you're correcting for significance because of the number of hypotheses tested, right?
Yes.
So in other words, these guys at the top are, you know, perhaps a very significant condition upon how many you've looked at.
Yes.
So
so you're talking about this little peak,
yes,
right?
Yeah, so I'm curious about that rest of them. So individually, they're much less significant.
Well,
so my question is, how much of the good stuff lies in there, basically?
I think this is kind of, yeah, we debate back and forth whether we prefer to do the early stopping method or the weighted FDRC method, because you're right, using weighted FDRC, we do lose a number of significant interactions. Those are of nominal lower significance than the ones that we pick up along the way. So the point of doing continued testing and sort of exhaustively testing all pairs and using weighted FDRC is that there might be a few here that are very highly significant that were just surprising, that our prior model did not think would be epistatic, and we're able to pick them up by doing complete testing, while we are in fact losing some of the ones that had lower nominal significance but higher prior probability of epistasis.
My second question is about the hubs. So when you do a random walk, you're going to be hitting those hubs a lot, regardless of where you start. My question is, if you use a null model of randomly starting the network in order to ask how much more, you know, do I identify individual nodes, then do the hubs go away? I mean, would that be an appropriate control?
That would be an appropriate control. So you're kind of combining 2 parts of our method. One way I'll address it is by saying that we get very different hubs for different traits. So I don't think that we're simply picking up protein-protein interaction hubs, for instance. However, you will overall have higher connectivity between any given gene and a PPI hub. And that definitely could influence our results, but I'm not sure that that's necessarily a bad thing. Because,
no, I mean, I'm
biologically,
not saying it's a bad
those are
thing,
interacting with more genes.
but it could still hold even after you correct for that. In other words, you know, right now you're sort of wondering, well, it sort of makes sense biologically, but perhaps it could be an artifact of the you know, random walks. If you did this correction and then the hubs are still significant, then it could refer you further.
I guess the only thing that I will say to address that is we actually attempt to correct for total degree in our definition of graph kernel. So we use a normalized Laplacian, which tries to take into account the number of neighbors that each node has and not overemphasize the hubs. But minor effects of that could still be in our data.
But I think that simply correcting for, and basically the effect of high degree on random walks is much higher than the degree would suggest.
Yes. Okay.
Great.
So I think that's a good suggestion. Thank you.
Very excited.
So we have time for one more question. Maybe we'll take a question from the back mic, if that's okay.
So your model is basically a blender, right? Can you put SNP gene interaction hubs in there?
SNP gene interaction.
Yeah, can you look at SNP gene? Yeah, basically.
Can we put them? Can we hope to get output that is SNP interaction?
Can you add, if you add the SNP data, will you get something interesting?
Oh, okay. So actually, our type 1 diabetes data is all SNP data, and I showed gene level results only because it simplifies the presentation, and we associate every SNP with a particular, actually with a set of genes that are near it. I'm not sure actually if that addresses the question that you're asking. Are you asking if I could combine the SNP level data with gene level data and find interaction between one SNP and an entire gene?
Yes.
Okay. I think that you could if you were willing to define your genetic variants at a gene level in a coherent way, which we have not looked at trying to do. But if you could do that, then yes, you could throw it into a model like this.
Thank you.
Thank you.
Okay.